Alright, Assalamualaikum everybody. How are you all? I hope that you all are doing well. Welcome or welcome back to my YouTube channel. So in today's video, we will be looking at AS and A-level biology paper 1, which is of the year May-June 2025, paper 1-2, and this will be specifically the part 2 of this yearly past paper, because we have already done question 1 to question 20 in the part 1, which the link of the part 1 you guys can find it in the description box below. So you can go and watch question 1 to question 20 over there. Now we will be starting off this class with question number 21 over here. So this is question number 21. Let's give this question a read. Question 21 over here says that the diagram shows the arrangement of the chromosomes with attached spindle fibers during a stage of mitosis. Which row correctly identifies the stage of mitosis and describe what is happening? So in this stage of mitosis, you guys can see that the chromosomes are aligned at the equator of the cell, right? And what is the phase in which the chromosomes are aligned at the equator of the cell, or you can say in the middle of the cell? So it is the phase of the metaphase, right? How many phases do mitosis have? Mitosis have prophase, metaphase, anaphase and telophase. And in prophase and in metaphase, what happens is that the chromosomes are aligned at the equator of the spindle, and you can remember it by this M for metaphase and M for middle as well. So you guys can remember that in metaphase the chromosomes are at the middle of the cell, also known as the equator of the cell, right? So the stage of the mitosis is the metaphase. The description is sister chromatids are replicating prior to the separation. Chromosomes are being positioned along the equator of the cell. So option C over here is the correct answer, because we know that in the metaphase the chromosomes are aligned at the equator of the cell. So hence this is option C is the correct answer for this MCQ. Now moving on to question 22, all of you. Question 22 over here says that during which phase of the cell cycle does the DNA replication takes place? So we know that. Let's move on to the notes of the mitotic cell cycle. So basically the cell cycle is divided into the G1 phase, the S phase and the G2 phase, and then there comes the mitosis phase and then the cytokinesis. Now basically they are saying that during which phase is the DNA replication taking place. So DNA replication is taking place during the S phase of the cell cycle. So hence the correct answer over here would be option D, the S phase. In the S phase the DNA replication takes place. Now question 23 over here says that human body cells in the interphase have 43 chromosomes. Now what is the interphase? G1S and G2 phase. The combination of this phase is known as the interphase. Now they are saying that body cells in the interphase have 46 chromosomes. Now we know that the human somatic cell have the 46 chromosome, which means that it is in a phase before the S phase where the DNA replication occurs, right? So it is in a phase before the S phase. Then it says that what is the correct number of the telomeres present in the prophase of a human body cell? What is the correct number of the telomeres present in the prophase? Now in the prophase, basically what will happen that the chromosomes will be duplicated, right? Now there will be 46 chromosomes, but they will consist of the sister chromatids as well. There will be 46 chromosomes, but they will consist of the sister chromatids as well. So there will be one chromosome will be consisting of the two sister chromatids, right? So if they are talking about the prophase, so we know that in the prophase the DNA replication would have been occurred, right? So and each chromosome will be having the one more sister chromatid, which means that it will having the two chromatids, right? So if the 46 chromosomes are having and each chromosome is having two sister chromatids, then the total of the chromatids we will be having is 92. So we are having 92 chromatids, and if we are having 92 chromatids, so we know that each chromatid has one chromatid has two telomeres, right? It has two telomeres. because telomeres are at the end of the sister chromatids. They are found at the end of the sister chromatids. So if one chromatid is of 2 telomeres, consisting of 2 telomeres, then how many telomeres would 92 chromatids consist of? So the 92 chromatids would consist of 92 into 2 you will do, and you will get 184. So hence the correct answer over here would be option D, that there will be 184 number of chromosomes, as there is one chromosome at the end of the 92 chromatids, right? When the DNA replication will take place, the number of chromosomes will stay the same. What will happen, that the sister chromatids are going to replicate and join the existing chromatid. This is what is going to happen, right? I hope that this is clear to all of you. Even if it is not clear to you, then I would recommend you to go and watch my playlist of the mitotic cell cycle P1, right? Topical P1 that I solved. Over there I have explained this concept in a lot more detail, right? So yeah, this was about this question. I hope that it is clear to all of you. Moving on to question 24 over here. Question 24 over here says that sometimes hydrogen bonds form temporarily between the bases of two strands of the RNA. How many hydrogen bonds form when the guanine and uracil each binds to their complementary bases? So basically now they are saying that how many hydrogen bonds are basically found between the nitrogenous bases. So we know adenine binds with the thymine, right, and the guanine binds with the cytosine. Now this is for the DNA, right? If I talk about the RNA, then in the RNA the adenine basically binds with the uracil and not with the thymine. And how many hydrogen bonds basically these are making? So over here you guys can see in my lecture notes that the adenine is making 2 hydrogen bonds with the thymine and uracil, and guanine is making 3 hydrogen bonds with the cytosine. The adenine and the guanine are the purine bases, which means that they are the double ring bases, and the pyrimidine bases are the thymine and the cytosine, which are the single ring bases over here. So we saw that the adenine to thymine there are 2 hydrogen bonds, and guanine to cytosine there are 3 hydrogen bonds. Now they are saying that when they will bind to their complementary, right? So guanine is going to bind with the cytosine and it is going to make 3 hydrogen bonds, right? And then the uracil is going to bind off with the adenine and it is going to make 2 hydrogen bonds. Hence the correct answer over here would be option B. I hope that this is clear. Now move on. Now let's move on to question 25 over here as well. Now question 25 over here says that the table shows all the possible DNA triplet codes for some amino acids. Now they are saying that these are some of the DNA triplets which are basically coding for the simultaneous amino acids. The amino acid sequence is shown: methionine, isoleucine, cytosine, tryptophan. What is the maximum number of different DNA sequence that could be produced, that could produce this amino acid sequence? Now basically if I talk about the methionine over here, so we know that methionine is basically formed by TAC, right? This is what is forming the methionine. Now for the methionine, we just have one DNA triplet code which is coding for the amino acid of the methionine. So methionine, only one code is there. If I talk about the isoleucine over here, so there are 3 DNA triplets that are basically coding for the isoleucine. There are 3 DNA triplets which are coding for the isoleucine. If I talk about the cysteine over here, so in the cysteine there are 2 DNA triplet codes that are coding for the amino acid. There are 2 DNA triplet code that are coding for the cysteine. Then if I talk about the tryptophan, so in the tryptophan only the one DNA triplet code is coding for the tryptophan. Only one DNA is being coded for the tryptophan. So methionine and tryptophan will remain same. Now let's talk about that how many different sequences we are going to have for the iso and the cysteine, like how many different DNA sequence could produce this whole amino acid sequence. Now for the methionine we will take this code TAC, right? For the isoleucine, for example, in one DNA sequence we take TAA, then we take the cysteine as ACA, and then the tryptophan is ACC, right? Now because we are only having one code for the methionine and the tryptophan, it will not change, but we are going to change for the isoleucine. We will now take another code for the isoleucine, and for the cysteine first we will take this because we have to make the possibility, and then for the third we will do TAT and then ACA, which is over here cysteine ACA, then obviously tryptophan is just one, so ACC, right? So these are the three possibilities that are being made if we change the isoleucine and take the one constant cysteine. Right now we are going to duplicate this, right? And what we are going to do over here is that we are going to change the value for the cysteine. We are going to now change the value for the cysteine over here. We first selected ACA, now we will go with the ACG. ACG. Now there are a total of 6, 3 over here and 3 over here. Now there are a total of 6 different DNA sequences that could produce this amino acid sequence, right? Now there are a total of 6 different DNA sequence that could produce this amino acid sequence. So hence the correct answer over here would be option C. Now let's continue with question 26 over here. Question 26 over here says that which row is correct for transpiration. So if I talk about the proper definition of the transpiration, so it is the loss of water vapors from the leaves, right, mainly through the opening known as the stomata. Mainly through stomata, right? This is what is known as the transpiration. So it is the loss of water vapors from the leaf mainly by the stomata. Why the option A and B are not being considered? The evaporation of water from the leaf surfaces. So basically evaporation is not the correct term over here, because we know that the water basically evaporates from the spongy mesophyll cells into these air spaces, right? These are the air spaces. So water actually evaporates from the spongy mesophyll cells, and then it comes to these air spaces between the spongy mesophyll cells, and then it diffuses out of the plant cell by the help of the stomata, right? And in the process known as the transpiration. So this statement A and B are basically not considered as the appropriate statements, right? And then if I compare the comparison of the water potential. Now if you look at the comparison of the water potential, so we know that as the water is being diffused out of the plant cell towards the environment, right? So the outside environment may have the less water potential. The outside environment must have a less water potential, and the inside environment must have a more water potential, because we know that the water vapors are usually moving down the water potential gradient from a region of higher water potential to the region of the lower water potential. So outside air would be especially dry or the low water potential air, right, which is having less water vapors. So higher inside, the water potential is higher inside air spaces in the leaf than the air outside. So hence the correct answer over here would be option D. Moving on to question 27 over here. Question 27 over here says that which features of the companion cells and the xylem vessel elements make them suitable for their function. So if I talk about the companion cells, so companion cells are the specialized cells which are needed for the phloem transport, right? These are the companion cells and they are required for the phloem transport. For example, the loading and the unloading of the sucrose. Over here there is the loading of the sucrose and over here there is the unloading of the sucrose. So these are the companion cells. And if I talk about the xylem vessel elements over here, so xylem vessel elements are these, right? These are the xylem vessel elements, and these xylem vessel elements are basically adapted for the bulk transport of the water and the mineral lines by the process of the mass flow, obviously, because we know that the xylem vessel conducts the bulk transport of the water and mineral lines by the mass flow. Now let's read the features one by one. Transport content of the cells in one direction only? No, because we know that the phloem can transport the substances in both the directions. Cellulose walls provide the support. It is true, but it is not the key adaptation, right? It is not the key feature. Nuclei allow cell division, so it is not suitable for their function, right? They are asking specifically suitable for their function. Numerous mitochondria supply energy. Yes, this is correct for the companion cell. Why it is correct for the companion cell? Because companion cell needs the energy for the loading process of the sucrose into the phloem sieve tube element, because we know that mitochondria generate the ATP, right? And many mitochondria are needed to generate ATP for the active transport process, because loading of the sucrose in the phloem sieve tube is the active process, right? Whereas unloading is a passive process usually done by the facilitated diffusion, but loading is an active process and it requires ATP. So, and we know that mitochondria generates the ATP, so there must be a numerous number of the mitochondria to supply the energy for the ATP. Now let's talk about some of the features of the xylem vessel element. Now xylem vessel elements usually have no cytoplasm, so that they can have a unrestricted or a mass flow without any resistance, you can say, right? And it has the lignified cell walls as well. Now the lignified cell walls provide the support. Yes, this is true. Nuclei allow division. Wrong, because the xylem is dead. Gaps between the cells allow rapid movement. No. Absence of the cytoplasm allows the mass flow. Yes, this is correct, because we know that because there is the absence of the cytoplasm, so there will be least resistance for the water to go up the xylem vessel tubes. So hence the correct answer over here would be option D. I hope that this is clear to all of you. Let's now move on to question 28, all of you. Question 28 over here says that the diagram shows some root cells and the arrows are basically showing the pathway of water moving from the soil. So this is mainly the apoplast pathway from which because the water is basically moving through the cell wall. So as the water is moving through the cell wall, so it means that it is the apoplast pathway. Which statement is correct for the pathway shown by the arrow? So we have already determined that the pathway that is shown by the arrow is the apoplast pathway. Now they are saying that water will be blocked by a band of suberin. Water will move through the Casparian strip. Water will pass easily through the root endodermis. Water will pass through the plasmodesmata. So water will not pass through the plasmodesmata. Why? Because it is a apoplast pathway, and in the apoplast pathway water does not pass through the plasmodesmata, whereas in the symplast pathway it does. Now the water will be blocked if the apoplast pathway is being followed, right? If the apoplast pathway is being followed, so the water will follow the apoplast pathway till the endodermis, right? In the endodermis, what will happen, that there will be this Casparian strip over here, and this Casparian strip is basically made up of a band of the suberin, right? It is made up of the band of the suberin. It consists of the suberin. And what is the suberin? Basically it is a component which is impermeable to the water and the solutes in the cell wall, right? Because when the Casparian strip is reached, then the apoplast pathway is blocked, and then the water is forced to cross the plasma membrane so that it can enter the endodermis. So the symplast pathway has started over here. So we will over here eliminate this option that water will move through the Casparian strip. No, it cannot. Water will easily pass through the root epidermis in the apoplast pathway. The water will not easily pass the cell endodermis because of the Casparian strip. Water will be blocked by the band of the suberin which is in the Casparian strip, obviously. So hence the correct answer over here would be option A. Now let's move on to question 29, all of you. Question 29 over here says that the graph shows the diameter of a tree trunk at different times. Now they are showing us the diameter of the tree trunk which is at its highest at 12 midnight, right? 12 midnight it is highest, and then 12 at the daytime which is 12 noon it is the lowest, and then it is again highest at the 12 midnight. So we basically have to understand this pattern and then we have to identify the correct statement. So first of all, let's understand this whole pattern over here. So basically at the midday, we know that at the midday at the 12 noon the sun is at its maximum, right? So at the midday the transpiration rate, the rate of transpiration is the highest, and because the rate of transpiration is the highest, we know that the water is being pulled up via the xylem. And how the water is pulled up via the xylem? By the cohesion and the adhesion forces, right? And both of these forces combine together to create a tension force within the xylem vessel, and because of this tension force generated by the water in the xylem vessel, it causes the xylem vessel and the surrounding trunk to shrink slightly in size. Hence the trunk diameter also reduces because xylem vessels are in the trunk, right? So because there is the tension generated due to the cohesion and the adhesion forces in the xylem vessel, so it will shrink. The xylem vessel and the surrounding tissue will shrink slightly and the trunk diameter will be reduced. And why are they highest in the midnight? Because we know that at the night the rate of the transpiration basically falls, and if the rate of transpiration will be fall, there will be the less need to pull up the water through the xylem, right? And if there will be less need to pull up the water through the xylem, so there will be less cohesion and adhesion forces generated, and then there will be less tension. So if there will be less tension in the xylem vessel, then the trunk will refill, or it will regain its original diameter and it will expand again. So let's read the statements one by one and try to answer our correct statement. Try to find the correct statement. Basically, J shows the expansion of the trunk as the water fills the xylem during the transpiration. No, this is wrong because the trunk diameter decreases during the day when the transpiration is there, right? It does not expand. So this is incorrect. And also at the night time there is not much transpiration as well. K shows a reduction in the diameter of the trunk as water is lost from the phloem due to the translocation. Wrong, because it is from the xylem, not from the phloem. K shows the reduction in the diameter of the trunk due to the water held in the tension in the xylem. Yes, this is correct, because as there is the tension in the water in the xylem vessels, so that is why the shrinkage of the diameter of the tree trunk has been shrinked, right? And if I talk about option D over here, L shows the expansion of the trunk as the phloem tissue acts as a sink at the night. Wrong. This is the whole wrong concept, right? So hence the correct answer over here is option C. I hope that this makes sense and it is clear to all of you. Now let's move on to question 30 over here. Question 30 over here says that the plan diagrams of the transverse section of the two plant organs are shown. These are not drawn to scale. Which tissue contains the proton pump? We know that the proton pumps is generally the feature of the phloem, right? It is found in the components of the phloem. So over here this is showing the cross section of a leaf, and over here this is the cross section taken from the root. So T over here is the xylem, S over here is the phloem, Q over here is the xylem, and R over here is the phloem. So because the proton pump is contained in the phloem elements, right? So the correct answer over here would be R and S, which is in option C. So hence the correct answer over here is option C. Alright, so now let's move on to question 31 over here. It says that the diagram shows three types of blood cells which are all drawn to the same scale, X, Y and Z. Which row shows the characteristics of these cells? So सबसे पहले what we are going to do over here is that we are going to identify X, Y and Z white blood cells. That what are these white blood cells? So basically Z over here is the neutrophil, right? Why we have considered this to be a neutrophil? Because the property of the neutrophil is that it is having the lobed nucleus, right? So we are seeing a lobed nucleus over here. So that is why we have said that this is a neutrophil. Then if I talk about Y over here, so Y over here is the lymphocyte. Y over here is the lymphocyte. How do we identify Y as a lymphocyte? Because lymphocyte nucleus covers 90% of the cell space and almost all of the cell space has been covered up by the nucleus over here. Now if I talk about the Z over here, so Z can be the monocyte or the macrophage over here. Why? Because it is having a kidney shaped nucleus. If I move on to my lecture notes over here, so you guys can see that the macrophages or the monocytes are having a kidney shaped nucleus. Now they are saying that name of the blood cell X. So the blood cell X over here is the monocyte, right? It is not the lymphocyte. So option C and D are already eliminated. Now we will look for the correct options from the option A and B. Engulfs the pathogen by the endocytosis. So endocytosis, a type of the endocytosis is the phagocytosis. And in the phagocytosis, there are two types of the red blood cells that are involved. One is the neutrophils and the other one is the macrophages or the monocytes. So over here we are having our option as Z, which is the neutrophil, because Y is the lymphocytes and lymphocytes are not involved in the endocytosis. Okay, what the lymphocytes do is that they respond to the antigens, right? They release the cytokines and they also destroy the infected or the viral body cells by basically reacting with the antigens, right? So that is why lymphocytes will be our correct answer for this option, that each responds to one antigen is the lymphocyte. So hence the correct answer over here would be option B. Moving on to question 32 over here. Question 32 over here says that the graph shows the oxygen hemoglobin dissociation curve for the three animals, animal 1, animal 2 and animal 3. It further in the question says that the shape of the each curve is influenced by respiration rate and the affinity of the hemoglobin for the oxygen. What can be concluded from the graph? So if I talk about the graph over here, so this is for the animal 2, this is for the animal 1 and this is for the animal 3. Now if the graph is basically shifting towards the right hand side, which is this graph, if it is shifting towards the right hand side, it means that the partial pressure of the carbon dioxide is high. And when the partial pressure of the carbon dioxide is high, the affinity of the hemoglobin for the partial pressure of the oxygen is low, right? And when the affinity of the hemoglobin for the oxygen is low, so it will dissociate the oxygen. So this is when the right shift will occur, which is occurring in the animal 3. If I talk about the left shift over here, which is for the animal 1 over here, so left shift occurs when the partial pressure of the oxygen is greater. And as the partial pressure of the oxygen is greater, so there is more affinity of the hemoglobin with the oxygen and it favors or it promotes the dissociation of the carbon dioxide from the hemoglobin, right? So this is about the right shift and the left shift. Now let's read the options one by one. Animal 1 has a hemoglobin adapted to have the higher affinity for oxygen than animal 2. This is true because the left hand shift of the hemoglobin dissociation curve is when the partial pressure of the oxygen is high, and when the partial pressure of the oxygen is high, so the hemoglobin is better adapted to the oxygen, right? It has higher affinity for the oxygen, so hence the correct answer over here would be option A. Now let's understand the other options as well, that why they are not the correct options. Animal 3 has hemoglobin adapted to have the higher affinity for oxygen. No, as the animal 3 is having a right hand shift, so the partial pressure of the carbon dioxide is greater, which means that the affinity for the partial pressure of oxygen is less. Animal 3 has a hemoglobin adopted to maintain the lower respiration rate than the animal 2. So basically animal 3, they are saying, is having a lower respiration rate. Lower respiratory rate basically means that there is the less oxygen that is being used up, right? Because in the respiration the oxygen is used up, and as there is a lower respiration rate, so the less oxygen is used up, so the partial pressure of the oxygen is relatively high. Animal 1 has hemoglobin adapted to maintain the higher respiration rate than the animal 2. If there is a higher respiration rate, then obviously the partial pressure of the oxygen will be less and the partial pressure of the carbon dioxide will be more, because if there is more respiration rate, then more oxygen will be used up and more carbon dioxide will be produced. And we know that as the partial pressure of the carbon dioxide increases, so it is going to shift towards the right hand side, which would be for the animal 3, right? So we will say that is why. So with this, all the other options are eliminated and option A has been selected as the correct answer. Let's now move on to question 33 over here, which says that which statement about the mammalian heart are correct. Now they have given us a series of 4 statements and we have to select the correct statement. Let's read these statements one by one. The right atrium and the right ventricle contains the deoxygenated blood going to the lungs. Yes, this is true. Why? Because we know that the right side of the heart contains the deoxygenated blood and the left side of the heart contains the oxygenated blood. Now statement 2 says that the left atrium and left ventricle contain the oxygenated blood going to the pulmonary circulation. So this is incorrect. Left side of the heart receives the blood. It receives the blood from the pulmonary circulation. Receives the blood from pulmonary circulation and gives it to the systematic circulation, which is the rest of the body. Which takes the blood to the rest of the body. So this statement is incorrect. Then statement 3 says that the left ventricle has a thicker wall than the right ventricle and delivers the blood at a higher pressure. So yes, this statement is correct, because obviously the left ventricle, the walls of the left ventricles are thicker as compared to the walls of the right ventricle, because from the left ventricle the blood is pushed to the aorta, and aorta has to push the blood through the whole of the body, so it must require a higher pressure. So that's why the left ventricle have a thicker wall. Also, this is one of the theory questions as well, so you must know this reason also. The pressure of the blood in the aorta is less than in the pulmonary artery. So no, this is incorrect. The pressure of the aorta is greater than the pulmonary artery, because वो पीछे से ही एक ज़्यादा pressure में आ रहा है. The left ventricle is pushing it with a thicker wall, which means it is pushing it with a higher pressure. So aorta में blood is at a higher pressure because it has to go to all of the body. Pulmonary artery से it simply have to go to the lungs, right? So that is why the correct answer over here would be option B. 1 and 3 are the correct answers. Now let's have a look at question 34 over here. Question 34 over here says that how is most of the carbon dioxide transported inside our body? So there are 3 ways by which the carbon dioxide is transported in our body. One is by when the carbon dioxide is dissolved in the blood plasma. The second method by which the carbon dioxide is transported in our body is by the carb amino hemoglobin. Is in a form of carb amino hemoglobin, right? When the carbon dioxide basically combines with the N terminal amine group at the N terminal of the polypeptide chain of the hemoglobin. So basically that is this carbamino hemoglobin. And the third one is as the bicarbonate ions. Now if I talk about their percentages, that how many percent basically is dissolved in the blood plasma, so almost 7 to 10% is carried by dissolving in the blood plasma, and then 20% is basically carried by the as a carbamino hemoglobin, and 70% has been is carried as a bicarbonate ion, right? So most of the carbon dioxide is transported in the blood in a form of the bicarbonate ions or as the hydrogen carbonate ions. So hence option C is our correct answer over here. I hope that this MCQ is clear to all of you. Also, I need to tell you that whenever you are solving the MCQ paper, make sure that you revise and revisit your previous concepts as well. लाइक मेरी यही कोशिश होती है कि मैं आप लोगों को क्वेश्चन भी एक्सप्लेन करूं और मैं आप लोगों को बाकी का भी पूरा कॉन्सेप्ट दूं, ठीक है? सो दैट इज़ वाई आई ट्राई माई लेवल बेस्ट टू एक्सप्लेन द MCQ टू यू गाइस, ताकि इफ़ अगर इससे मिलता जुलता कोई क्वेश्चन आ जाए, फॉर एग्जाम्पल if you are appearing in October November 2025 और इससे मिलता जुलता कोई क्वेश्चन आ जाए, so you guys should be able to answer that question, ठीक है? so this is the best way to do paper 1 कि आप वो क्वेश्चन भी करें और उसके बाद आप रिवाइज भी करें और रीविजिट भी करें अपने वो सारे पुराने कॉन्सेप्ट्स दैट आर रिलेटेड टू दैट स्पेसिफिक टॉपिक, राइट? Also, if you go to my channel, so I have made 2 videos on the one is for the attempting technique of the A-level biology and the other one is the preparation strategy for the A-level biology. So you guys can go and check that out as well. These are very informative videos and I have disclosed a lot of top key points and the key tips to attempt and to prepare for your paper 1. Let's move on to question 35 over here. Question 35 over here basically says that which row shows the tissue that are present in the walls of the trachea and the walls of the bronchus. So we know in the air passages there is trachea, and trachea then bifurcates or divides into the bronchus, which has a plural known as the bronchi. Now they are saying that which features are present in both the trachea and the bronchus. So let's read the features one by one. Cartilage. So if I talk about the cartilages, so cartilages are present in the trachea as well as the bronchus. The trachea is having the C-shaped cartilages. The trachea is having the C-shaped cartilages, whereas if I talk about the bronchus, so bronchus is having the irregular shaped cartilages, right? It is having irregular shaped cartilages. So the cartilage is basically found in both the trachea and the bronchus. And if I talk about the ciliated epithelium, so yes, cilia is found in the epithelium of both the trachea and the bronchus. Smooth muscles is found in both. So hence the correct answer over here would be option A. Now let's move on to question 36 over here, which says that which factors maintain the diffusion gradient for carbon dioxide at the surface of the alveoli. We know that when we inhale the air, so the inhaled air when reaches the alveoli, the partial pressure of the carbon dioxide is less over here and the partial pressure of oxygen over here is greater, and the blood that is coming from the lungs is basically the deoxygenated blood, which is having a greater partial pressure of the carbon dioxide. Now they are saying that which factors maintain the diffusion gradient for the carbon dioxide. So we know that the deoxygenated blood is going to come and cross the alveoli, and then the carbon dioxide is going to diffuse into the alveoli and the oxygen will diffuse from the alveoli into the blood capillaries. So the diffusion gradient for the carbon dioxide at the surface of the alveoli is obviously maintained by the blood flow around the alveoli, because the deoxygenated blood is moving, right, and it is crossing the alveoli and then it is becoming oxygenated. So the blood flow around the alveoli definitely provides the diffusion gradient for the carbon dioxide, because the blood that is coming is having the low concentration of high concentration of the partial pressure of carbon dioxide, whereas the blood which is the air that is in the alveolus contains a low partial pressure of the carbon dioxide. By this the diffusion gradient is maintained. Breathing movement, exchanging air in the lungs. Yes, this is also maintaining a diffusion gradient of the carbon dioxide, because as we inhale, the inhaled air contains a greater partial pressure of the carbon dioxide, or greater concentration of the carbon dioxide, right? And when the gas exchange basically takes place and carbon dioxide moves from the capillaries, pulmonary capillaries to the alveolus, then we exhale the air. So the more carbon dioxide air is exhaled out, and then when we inhale, then the inhaled air, which is less in carbon dioxide and rich in oxygen, comes in. So yes, the breathing movement, which is the exchanging of the air in the lung, is also maintaining a diffusion gradient for the carbon dioxide. Thin epithelial lining of the alveoli. Now basically the thin epithelial lining of the alveoli is not contributing to the diffusion gradient, right? It's not contributing to the diffusion gradient itself, but it is contributing to the rate of diffusion, right? It obviously increases the rate of diffusion, but it is not contributing to the gradient, the diffusion gradient for the carbon dioxide, right? So hence the correct answer over here would be option A, 1 and 2 only. Alright, so now let's move on to question 37. It is a very lengthy question. So let's give it a read. That a student made notes about the two diseases, hand, foot and mouth disease, HFMD and Lymedisease. So there are two diseases, HFMD and the Lymedisease. HFMD is caused by a viral pathogen and is transmitted in a variety of ways, including the ingestion of the water contaminated with the feces, which is also known as the fecal oral route, right? And by the inhalation of the droplets containing the pathogen. And then the Lymedisease is caused by a bacterial pathogen and is transmitted by the bite of the infected insect. For example, when the infected insect is going to bite you, it is going to transmit the bacterial pathogen inside the body. Which statements make a correct comparison between the diseases the student wrote about, and cholera, malaria or tuberculosis? Now let's discuss the options one by one. Antibiotics can be used to kill the vector of the pathogen that causes the malaria and the vector of the pathogen that causes the Lymedisease. The vector of the the vector of the malaria is basically the insects, right? The female Anopheles mosquito. Though this statement is actually wrong. Why this statement is wrong? Because the antibiotic usually kills the bacteria. Antibiotic usually kills the bacteria and it does not kill the vectors. Like if for example it does not kill the mosquitoes, right? They are controlled by the insecticides. The vectors are basically, like for example the mosquitoes, they are controlled by the insecticides, or you have to take any environmental measures to control them. Then the next statement over here says that HFMD can be transmitted in similar ways to either cholera or the TB. Now this statement is correct. How this statement is correct? Because we know that the cholera can be spread by the ingestion of the contaminated water, for example as the fecal oral route which we told about, and the HFMD is also being transmitted in this similar way. Similarly, if I talk about the TB, so TB is also spread by the inhalation of the infectious droplets, and same is the route for the HFMD disease as well. So either way is correct. So hence B is known to be your correct option over here. Now let's have a look that why C option is incorrect. The cells of the pathogen that causes HFMD and the cells of the pathogen that causes TB are prokaryotic. Now this statement is incorrect. Why this statement is incorrect? Because HFMD is viral, right? It is viral, so there is no prokaryotic cells used. And TB is bacterial. And if the TB is bacterial, so obviously over here the prokaryotic cells are involved. Then D option is also incorrect. Why? Because the transmission of the TB and HFMD could be reduced by chlorinating the drinking water. So this is also incorrect. Why? Because obviously we know that the chlorination of the water helps to reduce the waterborne diseases. For example, the cholera, right? But some of the HFD HFMD are not the waterborne diseases. Like for example, TB is not waterborne, right? Which spreads by the water droplets, right? So hence the correct answer over here would be option B. Now let's move on to question 38. Question 38 over here says that which statements explain why antibiotics may not treat the cholera successfully. So cholera is an infectious disease which is caused by the pathogen or the bacteria which is Vibrio cholerae, right? It is a pathogen for the cholera disease and it is bacteria. Vibrio cholerae is a bacteria. Now they are saying that which statement explains why antibiotics may not treat the cholera. So a possible reason for this is that maybe the infectious organism has developed the resistance against the antibiotic. Maybe the pathogen for the cholera has developed resistance against the antibiotic. That's why the antibiotics are not being affected on the Vibrio cholerae bacteria and are not being useful to treat the cholera. So this statement can be correct. Now let's look at the other statements as well. Antibiotics cannot harm the viruses. Yes, this is true in general, but it is not relevant to the cholera because cholera is caused by the bacterial disease. So this statement is incorrect and non-relevant. Antibiotics do not affect the pathogens that are eukaryotic. This is also true, but again not relevant to the cholera because cholera is caused by the bacteria which are prokaryotic in nature. So hence the correct answer over here would be option B, one only. Now let's move on to question 39, which is the second last question of this paper. The statement describes the phagocytosis of the bacterium by a macrophage. So they have told us the 5 steps by which the phagocytosis of the bacterium is taking place by the macrophage. Now they are saying that what is the correct order for the phagocytosis. Now we know that सबसे पहले हमारे पास क्या होगा, that सबसे पहले हमारे पास there will be the recognition and attachment of the bacteria to the macrophage, right? So how there will be the recognition and attachment of the bacteria to the macrophage. It is possible when the receptors on the cell surface membrane of the macrophages bind to the bacteria, right? So this will be our first step where the receptors on the cell surface membrane of the macrophages will go and bind to the bacteria. Then what is going to happen? Then the endocytosis process, which is the phagocytosis, generally it will take place that the macrophage is going to engulf that bacteria and enclose it in a vacuole, जिसको हम लोग क्या बोलेंगे, जिसको हम लोग phagosome vacuole भी बोलते हैं, right? It is also known as the phagosome vacuole. That phagosome vacuole, this is for example if this is a macrophage, right, and this is the phagosome or a phagocytic vacuole which has the bacteria, right? Then what is going to happen, that the lysosome will come into play. Lysosome, we know what is a lysosome. Lysosome contains the hydrolytic enzyme. So the lysosome is going to come and it is going to fuse with the macrophages. When the lysosome is going to come and fuse with the macrophages, so what will happen, that because there are the hydrolytic and the digestive enzymes in the lysosome, so it will break down the bacteria, right? So the digestive enzymes will break down the bacteria. When the digestive enzymes will break down the bacteria, so the pieces of the digested bacterium, what are they? So when the lysosome is going to break down the bacteria, it will be broken down into pieces, right? So when the digestive enzymes are going to break down the bacteria, what will happen, that it will break into the fragments. And macrophages, what macrophages will do, that it will do the antigen presentation, like they will display the antigens of that bacteria on the surface of the macrophages, right? So the antigen presentation will be there and this antigen presentation will help to activate other immune cells of the body, right? It will activate the other immune cells of the body towards this bacterium. So macrophage display the bacterial antigen on its cell surface membrane. So first step was 5 was the first step, right? Then the macrophages would engulf, then lysosome would fuse, digestive enzymes would break down, and the macrophage will display the bacterial antigen on its cell surface membrane. And due to this antigen presentation, the other cells of the body or the other immune cells of the body, basically they will activate, right? So the correct answer over here would be option C. I hope that this is clear to all of you. Now let's finally move on to the last question of this paper. This question says that two people G and H were each given an injection to protect them against a particular pathogen. The one person was infected with the antibodies, the other person was injected with a vaccine. The graph shows the concentration of the antibody against this pathogen in the blood of the two people G and H during a period of 20 days after the injection. Which row correctly describes the type of immunity shown by G and H? Alright, so basically the person G, you can see that in the person G the concentration of the antibody is being decreased, right, coming to the 15 day, whereas if I talk about the concentration of the antibody in the person H, so it took some time for the antibody concentration in the person H to increase to reach its maximum value and then to decrease. Now they say that which row correctly describes the type of immunity shown by G and H, because obviously the G, it is having the high antibody concentration at the start, right? At the 0 day it was having a high antibody concentration, which means that the antibody was injected artificially in this person, right? The antibody was injected artificially in this person, right? And then if I talk about the person H over here, now if I talk about the person H over here, so person H may be the person who is basically injected with a vaccine. Now in the vaccine basically we don't inject the antibody, we inject the weak antigens against a particular pathogen inside a human body, weak antigen, and a human body fight against those antigen and produce the white blood cells, or you can say the B lymphocytes that produces the antibodies against that specific antigens and create the memory cells, right? So they both, obviously the G and H, they both are the artificial, because artificial immunity, it is not the natural immunity. Why it is not the natural immunity? Because they have been injected, right? If anything is injected, even if the antibody is injected or the antigen is injected, it is known as the artificial immunity. So we will cut off the natural option. Now it's about the active or the passive, that whether the immunity is active or passive. So if I talk about the antibody, so whenever the antibody is being injected, so it is the passive process, right? Whenever the antibody is injected, so it is a passive process, so it would be artificial passive immunity. And whenever the antigen is injected in the body, so it would be the artificial active immunity, right? So hence the correct answer over here would be option B. I hope that this MCQ is clear to all of you. Now with that we are done with the whole paper of May-June 2025, paper 1-2. I hope that this whole paper is clear to all of you. If you guys want to watch the part 1 of this video, you guys can go in the description section below and you guys can find the video link for the part 1 where we solved question 1 to question 20. So I hope that you guys understood each and every question I explained in today's class. If you guys did, then do give this video a thumbs up. Do tell me in the comment section below that how are you finding these lectures and which yearly past paper should I solve next for you guys. Right, so yeah, that was it for today's class. If you benefited a little from this class, even a little from this class, do remember me in your prayers. Take care everyone. Best of luck from my side for your upcoming examinations. Keep working hard. Allah Hafiz everyone.